Okay, folks, I think we'll get started. How's everybody doing? How's your studying coming along? I see some smiling faces. No, I see a couple no's too. I guess we have to have a diversity of opinions for this. What should I say? I'll do this like I've done the other ones, and if you haven't been to the other review sessions, I'll remind you that these are review sessions where I'm wanting you to have questions about things. A question is not just go over that again, because I will just point you to the video if you want to go over that again. So where you have questions about something or you don't understand something, that's really the aim of the review session. I have not yet written the exam, so now that I know everything that you guys don't know, I'll know what to go put on the exam. That's a joke. And it's a joke I've used every review session, I think. But maybe that's why I got no laughs this time. The format will be just like the previous two exams, as I said. It'll be about 150 points. Time shouldn't be a factor. I may change the distribution of points relative to each other on the different sections, as I noted in class. But that's really about it. I wanted to thank everybody for coming on Friday. It was a big day for me, and that was a fun day for me. So I hope that it was fun for you as well. I really had a good time with it, so it was pretty cool. That said, where should we start? Yes?
Can you explain the purpose of the lac promoter, since if it uses a lac repressor to bind to the starting site? What does the lac promoter do?
Okay, so the question is regarding the lac operon, which has several components, and then basically what does the lac promoter do? So let's take a quick look at that and see what I can do to help you with that. So the lac, gene is under gene expression, that's why. Okay, and so the lac operon, if we look at the operon region, okay, that really shows all the players right there. So the lac operon, I'll remind you that an operon is a collection of genes, all controlled by a single promoter. So that's what an operon is. We say that such an arrangement is polycistronic, meaning that it, what I just said, multiple genes, all in the same location under the control of the same promoter. The promoter is, what is the function of the promoter? The function of the promoter in the lac operon is exactly the same as the function of the promoter in any other prokaryotic system. That is, it's the place where the RNA polymerase binds adjacent to and starts transcription. Now, it seems like it's different because of the involvement, as you asked, about the lac repressor. So the lac repressor binds to this region called the operator, and the operator region is immediately adjacent to the promoter. So by the binding of the repressor to that area, it affects the ability of the RNA polymerase to bind to the promoter. So since there is this effect, this can actually block transcription because it physically blocks the RNA polymerase from binding to the promoter. One thing about the promoter that I noted was that the promoter for the lac operon is a fairly weak promoter. It doesn't have very close sequence agreement with the consensus promoter sequence that I showed you before. That is the one that has the ideal sequence for a strong promoter. And that means that this cap site that's right here plays a very important role, because the cap site is the binding for the cap protein, and when that binds to the cap site, then you have increased transcriptional efficiency and overcomes that limit with respect to the weakness of the lac promoter. But the function of the lac promoter is just like any other promoter, to have a place for the RNA polymerase to bind and transcription to begin. Long answer to a short question. Yes?
Can you go over riboswitches and specifically like how much we need to know about those? Because it kind of just seems like they can bind a molecule and they can do one thing or the other thing, but it can do both.
Okay, so basically, if I understand the question, it is what are the considerations for riboswitches? And actually, I jumped back here. Riboswitches being here. All that a riboswitch is a region of RNA within a messenger RNA that can bind to a molecule. So I've shown you how in, for example, prokaryotic transcription, that you can have factor-independent sequences in a messenger RNA that favor the transcriptional termination. And that happens because of a structure in the messenger RNA that forms that kicks the RNA polymerase off. So all this is, in the case of a riboswitch, is something that is another structure. That other structure is now capable of doing something besides kicking off the RNA polymerase. This other structure is capable of binding to a molecule, and it's the result of binding to that molecule that has the effect of either terminating or anti-terminating transcription. So it can go either way. We can have the binding of the molecule favors the formation of a terminator, in which case the transcription will stop at that point. Or you could have, in a different gene, you could have a riboswitch that binds to a molecule and stops termination. In other words, it allows transcription to occur. So it doesn't really matter for the purpose of what the outcome is. The purpose of the riboswitch is that it allows input of the cellular environment to controlling transcription of a gene. So if you had, for example, a riboswitch that had the ability to bind lysine, and the riboswitch was located in front of a lysine operon, then you would predict, based on what I've told you about riboswitches, that lysine binding to a riboswitch would favor a termination sequence, because when lysine is abundant, that's the only time it's going to bind, and when that happens, then termination of the operon would cease. Okay? Whereas if you had some molecule that, let me think about a good example where you would have anti-termination. Let's say that you had lactose binding and you had a riboswitch for lactose and you had the lac operon. You only want that sequence to continue to transcribe, the polymerase to continue to transcribe if you had the lactose present, and so in that case you would predict that it would be an anti-terminator that would allow that to go forward. Does that make sense? So you can envision it going either way, and I haven't given you any specific examples. I just want you to understand the phenomenon and what a riboswitch is capable of doing. That's the important part of a riboswitch. Yes?
Just to clarify, is that controlling transcription or translation?
Okay, so it's not controlling either. It's controlling transcriptional termination, right? So transcription has already started because that's how you have this RNA here. So it's not controlling transcription initiation in any event. It's controlling transcriptional termination, right? Yes?
Are we going to need to know the specific like bird units for like the RNA, like the RNAs and prokaryotic and eukaryotic cells? Like the specific numbers for each of them?
Yeah, that's a good question. I would say in general, no, but certain ones that I've talked about that have specific names, I think are important. So particularly the 16S ribosomal RNA, which has the Shine-Delgarno sequence, is one that I've talked about a lot. And the other one that I've talked about a lot was the 23S, which has the ribozyme activity that makes the peptide bonds. So certainly those two, yes. But I'm not going to ask you to say what's the eukaryotic equivalent of the 23S or any of that, because I think that's just needless memory work. Yes?
Can you explain the difference between a sequence element and a conserved sequence?
Okay, so can I define the difference between a sequence element and a conserved sequence? It's a very good question. A sequence element is simply a sequence. So we put the term element beside it because when people talk about sequence elements, they're usually talking about a little short stretch of sequence, and that little short stretch of sequence is embedded in the enormity of a chromosome. Okay? So a sequence element is just a specific sequence. That's really all it is. A conserved sequence is something that you observe as a result of comparison of multiple sequences from, say, different organisms. So the conserved sequence, for example, that you saw for by the way, and a common question I've also gotten to this is conserved sequence versus consensus sequence. So you can largely think of those two as interchangeable, okay? I showed you a consensus sequence for the TAT-AAT sequence of the promoter of prokaryotic cells. You see a consensus sequence because there is conservation of the sequence. So what is conservation? Conservation means that it's non-variant. I won't say it's invariant, but it's not very variant. Okay? So to give you an example, if we look at the consensus sequence for a prokaryotic promoter, it's TAT-AAT. And that's because when you average up all the various promoters, you see something like 70% T in the first place, 70% T in the A in the second place, et cetera, as you go through. That's far greater than you would see if you had random sequence. Because if you had random sequence there, you would expect to see 25% of the time A, 25% of the time G, 25% of the time C, 25% of the time T. Okay? So there's a conservation, meaning that there's a bias in terms of one sequence, and you see that conservation as a result of aligning all these up, and then you say, oh, here's the consensus that we see from that. So if you want to think about consensus and conserved sequence in the same sense, you won't be wrong. But in terms of what a sequence element is, that's something that's completely different. It's just a small section of sequence is all it is. A sequence element could be conserved. It doesn't have to be.
Okay. Because I just know that the Tata box can be included in sequence element.
A Tata box could be described as a sequence element, certainly.
But it's also in a conserved sequence?
So a Tata box is a conserved sequence. So there's an example where you could call this a sequence element that is conserved. Yes.
Okay.
But you might have another sequence element that is not conserved.
Okay.
Yeah.
Like the GC box or something?
Yeah, something like that.
Okay.
Right. Good. Clear as mud. Yes?
Is the Shine-Filgarno sequence used instead of promoters, or are there also promoters?
No, Shine-Filgarno sequence has nothing to do with promoters. So Shine-Filgarno sequence is a sequence in the messenger RNA to align the ribosome so it knows where the translational start is. Has nothing to do with the promoter.
Okay, so it's just for mechanically
Mm-hmm.
aligning it?
It's the Shine-Filgarno sequence that forms base pairs with the 16S, but that's a phenomenon that's going on in translation. Remember that transcription has already occurred by that point, so it can't be a transcriptional phenomenon at all. Okay? Yes?
So the Shine-Filgarno sequence is in the mRNA?
The Shine-Filgarno sequence is in prokaryotic messenger RNAs. Yep. Yes?
Is the corresponding part of it that's in the ribosomal RNA, is that also called the Shine-Filgarno sequence?
Yeah, so is the complementary sequence that's in the 16S ribosomal RNA also, does it have a name of its own? And the name I would give it is the complement to the Shine-Filgarno sequence. So no, it doesn't. Yes?
Is it similar to a Kozak sequence?
No. So is the Shine-Filgarno sequence similar to a Kozak sequence? No, it's not. So a Kozak sequence is not very conserved. If you look at that region I showed you, there's only 2 or 3 bases around the start site that appear to be where there's a bias, and they have no relationship whatsoever to the TAT sequence. So no, there's no relationship between those at all, other than to say that they both appear to be conserved sequences near a translational start site. But other than in terms of the sequence or function or anything, there appear to be no relationship at all.
Is its function?
The Kozak sequence? Yeah, so the Kozak sequence, people think, has something to do with helping to identify a start codon for translation. So in that sense, it functions like a Shine-Filgarno sequence, but the difference is there's no equivalent structure in the ribosomal RNA that allows that alignment. So the fact that there's a bias for those bases that I showed you suggests it has function, because that's whenever we see conservation of sequence, we usually associate that with function, but it's a weaker association, definitely a weaker association. Yes?
Explain the difference between exon shuffling and alternative splicing.
Yeah, what's the difference between exon shuffling and alternative splicing? Exon shuffling will use alternative splicing, okay? But people use the term exon shuffling for a couple of different things. So let me just tell you the two ways in which people use the term exon shuffling. So exon shuffling, as I use that term, really has to do with the alternative splicing phenomenon. Let's imagine that I make, let's pull up an alternative splicing figure. I think it's in the transcription one. Here we go. Splicing, maturation, splicing, alternative splicing here. Okay. So the way I use the term is, as you can see here, you have a series of codons that all could end up in a mature protein. And alternative splicing says, well, we're going to take this one, and this one in the final protein. That is, we're not going to take this one, we're not going to take this one. So I splice the end of this one to the end of this one, the end of this one to the end of this one. Okay? That would be different than if I said this, 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 this. Okay? So the alternative are just two different ways of splicing that same thing. And that's the way I actually use the term exon shuffling. People also use the term exon shuffling to describe evolutionary shuffling. So over evolutionary time, you might say, well, okay, this guy right here has a function. One of the things we see with exons is we sometimes see specific functions that are associated with just that sequence. So let's imagine, for example, that I had this sequence that was able to bind to ATP. Okay? So this sequence is able to bind to ATP. Exon shuffling, as it's used in an evolutionary sense, would indicate that this sequence is not only associated with this protein, but it's associated with another protein on a completely different chromosome that also needs to bind to ATP. Well, how did that happen? It happened by virtue of the fact that over evolutionary time, this sequence somehow got duplicated. And as it got duplicated, it itself got shuffled from one place to another. That's why they use that term of shuffling. That term of shuffling is different than this shuffling. This is real time. This is happening right now. Whereas the other term that people use to describe that I've just told you is over evolutionary time, which is a much longer time span. Does that make sense?
So you said in alternative splicing, you can't, like the blue couldn't go before, or the blue couldn't go before the red, but for exon shuffling over evolutionary time.
Over evolutionary time, you could have any kind of movement. That's correct. But you would never see exon 3 appear before exon 1 in real time. Okay? Because the order is going to go this, 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 this. And so the only shuffling you can do is popping out any of the ones, here. Or I could even start with number 2, for example. But I could never have a protein in real time that would have 2, 1, 3, 4, for example. Because that's just not the way the splicing system works. Make sense? Yes?
This is kind of a question about old stuff, but in one of my other classes it came up that methotrexate helps for ectopic pregnancies.
Mm-hmm.
So I looked it up, but it also said it does abortions, so how does it do both?
Well, I'm not sure I can answer that question for you. But I will tell you that methotrexate is used for other purposes, and it probably has to do with its ability to restrict cellular growth. But I wouldn't be qualified to answer that question. I'm sorry.
In the initiation phase
Yes.
of transcription,
Yes?
no, translation.
Okay.
The FMET is, can you just clarify what step happens first, if the 16S lines up with the Shindell-Garno, or if that 1T with the FMET goes first?
Okay. Let's pull up the figure. Okay. So the initiation of translation, and basically want to see the steps involved in the initiation of translation. So let's see there, transcription, translation. I've got to align those things, don't I? There we go. Okay, and so let me think here. Was it here? No, I want to say it's here. No. Oh, here we go, I'm sorry. Well, we're getting closer. pairing initiation. This is the thing that you're asking about, right? Okay, so you notice that you have basically at this point the FMET, which is coming in with the small subunit of the ribosome and the messenger RNA. If you want to think about these three things all together, that's fine. There actually is more of an order to that, but those three all at that point would all be equivalent as far as I'm concerned. Yes?
My question is about UTRs.
Uh-huh.
I remember hearing about them, like in regards to like polyadenylation of
Mhm.
the polyadenylate,
Mhm.
but I couldn't really find much else on them. So I'm wondering if you can kind of clarify their significance.
Yeah, that's a really good question. It's one that I'm not going to be able to answer fully, partly because we don't fully understand what happens with them. Let's think about what is a UTR. So UTR is an untranslated region of a messenger RNA. And when we look at eukaryotic messenger RNAs in particular, we find UTRs on the 5' end of the messenger RNA, and we also see them on the 3' end of the messenger RNA. Many of them appear to play roles in gene expression, partly by the function that you mentioned with respect to their role in polyase, and partly with their ability to affect the stability of a messenger RNA. You could also imagine that some of the UTRs, and this is actually known, some of the UTRs play roles in binding to things and affecting the gene expression as a result of that. So when I showed you, for example, the iron metabolism, and I showed you the IRE is a UTR, and it's able to bind, it's able to be bound, in this case, by a protein. But that untranslated region is a target for the IRE binding protein and affects gene expression as a result of that. There are probably many things like that that either can be bound by proteins or in some cases can be bound by molecules if it's able to form a 3D structure that could bind to a molecule, in which case it's acting like a riboswitch. So those are two common things that you might expect to see associated with a UTR and a messenger RNA. Yeah.
I asked you this in an email, but just to clarify,
Okay.
On the central role of glutamate.
Yes.
Slide,
Oh, yeah.
is there both a glutamate transaminase and a glutamine?
Okay, so let's go back. I think I've got one figure that's got a mislabeled thing on it. And whenever I have a mislabeled thing, I'm not going to hold you responsible for my mislabeling, but I'll show you what I think you're referring to. Let's go back here. And do you remember which one it is? Is it this one?
No, I think it's before that.
Glutamine
I
synthesis.
think it's right after the glucose alanine cycle at the top.
This one? Okay. Yes, and so on this one I'm showing that's glutamine transaminase, that's right.
And I guess comparing that with the highlight, and the highlight when you were explaining what glutamine transaminase does, it sounded like you were explaining the glutamic acid to alpha-ketoglutarate reaction.
Well, now, so actually, no, I'm not, because this reaction right here is going down here. This glutamine transaminase is referring to glutamine to glutamic acid.
Yeah. I guess when I was reading the highlight about it,
Uh-huh.
it just sounded like you were describing the other reaction.
Well, while I've got you here, let me just take a look at that highlight and we'll see. Because somebody else has asked me the same question and we need to get that clarified. So let's see. Number 5. All right. Are central to handling ammonia in the body. Glutamic acid can be deaminated to form ammonia. Glutamic acid can pick up an ammonia, thus detoxifying it, yes, to become glutamine. That's glutamine synthetase. In cells, ammonia can also be added to alpha-ketoglutarate to make glutamic acid. That's correct also. That's in fact, I'll show you that in a second. In cells, the amine of glutamic acid can be transferred, I can't spell pyruvate. Enzyme equals the amine of glutamic acid can be transferred to there's the problem. It's actually right there. I thought the problem was on the figure. The problem is in the highlights. This should say glutamate transaminase. not glutamine. Yeah, so that's the problem. I was thinking it was on the figure itself. So it's not the figure. That's an easy problem to fix. But again, when I make a mistake, I'm not going to mess with, if you said that for the question I'd ask you, I would accept that as a correct answer.
So both of those enzymes do exist then?
Both those enzymes do exist, yes. Yeah. There is a glutamine transaminase. In fact, you can actually see it on the figure. So the glutamine transaminase that was there, hold on, here is right there, glutamine transaminase. So it's converting this. Well, actually, now hold on. So, oh, okay, no. So it is confusing because you have both this one, which glutamine transaminase can use. That is correct. But you also have the conversion of glutamic acid to alpha-ketoglutarate, which also will convert that. So this one's a glutamine transaminase. This one's a glutamate transaminase. That's the confusion. But it is stated wrong in the highlights. I'll change that. Yeah. Now everybody's totally confused. Yes?
Mine's a question about the difference between DNA polymerase 1 and 3.
Yes.
So I was wondering if processivity is directly correlated with the beta clamp?
Yes, very good question. So is processivity directly correlated with the beta clamp? And the answer is absolutely. It's what causes it. So you see the processivity, which is the tendency to stay on a DNA, requires the beta clamp. And since the beta clamp binds only to polymerase 3, that's why polymerase 3 has high processivity and why polymerase 1 has low processivity. It jumps around because it doesn't bind to the beta clamp.
Then is that feature of DNA polymerase 3 why it's so rapid as well?
That's a good question also. It may play some role in that, but that's not completely clear. Yeah. So the reason it's hard to sort out is when you look at the replication fork, you see it's really set up for a polymerase 3, and there's no equivalent thing that comes up for a polymerase 1. Polymerase 1 works on short gaps, and you don't really need to have all the gyrase, topoisomerase, helicase, and so forth, because it's usually typically very short stretches of DNA that it's working on.
So then are there any key differences other than the beta clamp and the processivity that we should know between DNA polymerase 1 and 3?
Well, anything I say in class, of course, always fair game. So I did talk about different activities associated with those, yes. So and different functions associated with those, yes. Yes?
Regarding RNA interference,
Yes?
is the significance of that bubble in the microRNA? Because it's not in the size of RNA, but it is in microRNA.
Yes. So the question had to do with the RNA interference, and specifically the microRNAs have a little bubble associated with them, whereas the silencing RNAs don't have that bubble associated with them. And I'm not sure if there was a question associated with that, but basically the difference has to do with the fact that if you think about it, the cell is, when it puts that little, the cell has to put that bubble in there. And it puts that bubble in there by virtue of the fact that it's transcribing a DNA sequence that has evolved over time to have that bubble. Okay, because remember, it's an RNA, and RNA is made from DNA, so that bubble is there because the DNA over evolutionary time evolved to have an extra base in there that, or two that gave that bubble, all right? Why? Well, it may well have to do with this is a little signal in the cell that says this is cellular as opposed to a perfectly matched sequence, which is what a virus is going to have. That's just a guess. But that's one possibility for why you see the bubble in one versus the other. In the end, the same system operates where you've got the Argonaut sequence that's coming in and cleaving and breaking the messenger RNA.
You mentioned something about it having to do with differentiating which strand to cut, was it?
So that can play a role in telling which strand to cut. That's correct. Because that's more efficient if you think about it. If it's random, which is what it is for a viral sequence, you've got a 50-50 chance of getting it. Whereas the cell has no way of knowing which strand of the virus strand is the one it wants to knock out. It has no way of knowing that. So a 50-50 chance is good. Whereas in the case of the cellular DNA, it does know which one it wants to knock out because it knows which one the gene is. So you put, let's say you put the bubble on the other side of where the gene is, bang, now you've said, okay, destroy the top bubble strand and leave the other strand intact so that you can pair. Does that make sense? That's more detail than I've gone into in class, so if you haven't heard that before, don't sweat that. Yes?
To clarify, is the iron response element 1 mRNA for both, to translate to both ferritin and transferrin, or ferritin and transferrin?
I'm not sure I understand the question.
So the iron response element is involved in the translation of ferritin and transferrin.
Well, no. So let's go to the iron response element. Let me show you that. So one of them plays a role in translation. One of them plays a role in stability of message for after something has been transcribed. So they're different phenomena. Okay, and the iron response element. Okay, so when we look at ferritin, okay, so ferritin is the protein that carries iron, and the cell wants to make abundant ferritin when it has abundant iron, because it's got to gobble up that iron and keep it from forming reactive oxygen species. So when iron is high, it wants to be making ferritin. Right? Well, iron binds to the iron response element binding protein, and when it does that, it cannot bind to the iron response element. So high levels of iron will prevent the binding here, and it's the binding here that stops translation. So if you're preventing the stopping of translation, you're favoring translation of that. So it's here where translation is controlled. That's not the case when we look at the transferrin receptor. The transferrin receptor, on the other hand, has several iron response elements at the 3' end of the sequence. There's no involvement of translation. So the translation, if this thing were left completely alone, translation is going to occur just fine. The only difference here is that when you have no iron, okay, then the iron response element binds to these, the iron response element binding protein binds to these things, and that's what protects the message from getting degraded. So low iron will favor the formation of the receptor, which is going to help bring in more iron, all right? High iron is going to say, we've got plenty of this message, go ahead and destroy this with nucleases. But that has nothing to do with translation. Okay? You look puzzled.
So they're both iron response elements, but they're both just called iron response elements.
Well, they both do the same thing. Okay? So the same thing is very simple. They both bind to an iron response element binding protein. The effect is different. In the first case, the effect is to prevent or allow translation. Here, the effect is to prevent or allow messenger RNA degradation, right? So the effects are different, even though the function is just simply binding an iron response element or not binding an iron response. an iron response element binding protein or not binding an iron response element binding protein. It's a mouthful. Make sense? Yes?
Just a really quick question.
Okay.
Just to clarify, nucleosome is a histone instead with DNA wrapped around it.
Okay.
I found like a couple diagrams and that's what it's
Okay. So the question is really, what is the definition of a nucleosome? So what you said is partly right, that a nucleosome is a section of DNA that's wrapped around a histone octamer. And the difference between a nucleosome and a core particle is that a core particle doesn't have the flanking DNA sequences hanging off of it. That's really the only difference. So the core particle would have simply that circular region, but no other DNA tail hanging off of it. Whereas when you have the DNA tails hanging off it, that's what you call a nucleosome. Sure. What was that? I thought it was like a weirdest phone signal or phone ringtone I'd ever heard. Yes.
It was just related to what you were just asking.
Uh-huh.
What did you say was the product when it didn't have the tail?
So it's called so the question is, what is a nucleosome without the DNA tails? And that's a core particle. Core particle is the most fundamental element. Yes.
Okay, this is relating to translation.
Okay, relating to translation.
The first step kind of as that activation with that like charging of the tRNAs.
Mm-hmm.
So is that just always happening
Okay, good question. So the question has to do with the charging of tRNAs by the aminoacyl tRNA synthetases, also known as Kevin, right? So the question was, is that happening all the time? And if so, or even if not so, where is it happening? I think that's safe to, okay. So yeah, that's happening all the time. That's really got to happen all the time so that translation can proceed without interruption. And translation is going on almost all the time. And yes, that process is going on in the cytoplasm because that's where translation itself is going on, is in the cytoplasm. Yeah, and by the way, there is, not to confuse the subject, because you mentioned mitochondrial matrix, I'll say something about that also, but you're not responsible for this. Mitochondria actually do translation on their own inside of them, and mitochondrial translation is interesting. Now, as I'm saying this, I'm misstating this. It's not the translation; it's actually the transport of proteins into the mitochondria, and that happens from translation. And that is a function of what's called a signal sequence. So I talked about signal sequences that target things for the endoplasmic reticulum. So a signal sequence on the end of a protein can get it targeted for inside of the mitochondria. Yeah.
Are those, sorry, side question. Are those signal sequences, those are clipped off, right, once they basically...
The signal sequences get clipped off once the translation gets going, yes. Okay. Yes?
So we talked about attenuation with the tryptophan
Yes.
operon, but is that something that happens in the synthesis of all amino acids and we just have tryptophan as an example?
Yeah, that's a good question. So the question had to do with attenuation of the tryptophan operon, and do all amino acid operons have that? The answer is they don't all have that, no. But there are several in E. coli that do. And I think I may have mentioned valine or leucine, I think, are a couple. But you're not responsible for anything except the tryptophan one. The reason I mentioned that others do it is because you can see a sort of a commonality of that the effect of translation on transcriptional termination might be a useful function for control of other genes, and in fact it is. Yeah. Yes? Yes. Okay. Okay, so the question is, how does attenuation affect things? How does it slow down and so forth? So let's take a look at this figure right here, which shows the 4 regions of interest for attenuation, region 1, 2, 3, and 4. And they have 2 different scenarios that can happen. So if you have a high level of tryptophan, you can imagine that the ribosome is going to have nothing to slow it down. Once it gets started, it's just going to go moving along, smoothly translating, whereas if it's low on tryptophan, it's going to get stuck when it hits tryptophan things. Okay? So the fact that there are in this region, there are codons that specify for tryptophan. There's more than one of them. There are several that are there. What they're doing is, if there's low tryptophan, it's slowing the ribosome down. So slowing the ribosome down allows for this structure to form, okay? And, I'm sorry, slowing it down, it allows for this structure to form, which prevents the termination, right? So if it slows down, it gets stuck in one, waiting for that tryptophan tRNA to come in. And you might say, well, if it's low in tryptophan, it doesn't come in. Well, these are all relative things, right? So the cell always has tryptophan. The cell always has tryptophan tRNA. The question is, does it have high levels, or does it have low levels? If it had no tryptophan at all, the cell would be dead. All right. So, but with low levels of tryptophan tRNA, this guy slows down and covers up this. It doesn't make it up here to the region 2. So it's covering it up. It doesn't make it to region 2. So region 2 can pair with region 3, and that's what prevents 3 from pairing with 4. When 3 pairs with 4, as it does up here, then you've got transcriptional termination. That's a terminator sequence, kicks the RNA polymerase off, and the transcription of the operon is halted. Does that answer your question?
Is that like specific, like, does it occur at a specific sequence,
It does occur at a specific sequence because it's actually in this region. You can see where it says TRP codons. It occurs right there at that region because it's the absence of tryptophan that's essential for that. And that's why these codons appear in this leader region. The peptide that's made here has no function in the cell whatsoever. No function. The cell doesn't care what the coding of this protein is, other than the fact that these tryptophan codons slow the ribosome down if there's low tryptophan. That's the only function that's of that in the cell. Yes?
And then just to clarify, so you have transcription starts at the tryptophan operon, and then translation will start right after it crosses the AUG of the leader sequence. So that's why the break one after the other.
I'm not sure I understand your question. Can you repeat it?
So transcription starts and then translation is right behind it.
So transcription of the tryptophan operon starts, and then because we're in prokaryotes, translation starts almost immediately right behind it. That's correct. If that's your question, yes. Okay, that's fine. Yeah, sure. I just want to make sure I was answering what you were asking. Yes?
So in lecture, it seemed like you said that the initiation factor 2 and the elongation factor TU were almost synonymous.
Yes.
In the highlights, it seemed like they were separate, so
Okay.
I was wondering if you could explain.
Okay, so the question was, in the lecture I talked about IF2 and EFTU as being synonymous, and in fact they are quite synonymous. IF2 binds only to the FMET tRNA and protects it from being broken down into FMET and the tRNA. That is, it stabilizes that structure against water. And that's the same thing that EFTU does for everything else. So IF2 is very comparable to EFTU. It's IF2 and EFTU. I said IFTU. It's not IFTU. It's IF2. I do that all the time. IF2 and EFTU. Yes?
This is way back on the glyoxylate cycle.
Okay.
But I've written down that it's activated in high energy conditions. Does that include like high NAD plus concentrations or just if you have high acetyl-CoA?
Okay, so the question had to do with the glyoxylate cycle. So let me go back to the glyoxylate cycle and show you that. And the question was, does it only operate under conditions of high energy or under conditions of high NADH? Is that the question?
Yeah.
Kind of. Close enough, right? Okay, so let's pull up I thought it had it down here. There it is. That's not a very good one. Let's pull up this one. Okay, so the glyoxylate cycle is shown here, and it's shown superimposed on the citric acid cycle because of the relationship between the two. In the glyoxylate cycle, what you have is a short circuit of the cycle where isocitrate is broken down into succinate and glyoxylate. So the question had to do with high energy versus high NADH, and the answer is that they're sort of synonymous. Generally, cells are going to have high NADH when either the cell is not doing much, okay, or if the cell is running out of oxygen. That would be bad, right? But remember that the glyoxylate cycle is occurring in plants which don't have muscular contraction, so they don't have widely varying needs for that, and also in bacteria. And bacteria have a way of adapting to and adjusting for that, okay? So the plants are the main concern as far as the energy goes and the NADH concentrations. Why is it relative to the high NADH concentration? Well, keep in mind that what the glyoxylate cycle is doing is it's bypassing 1-2 reactions that require NAD. So consequently, by going the glyoxylate cycle, the cell has much less need for NAD. It can make intermediate oxaloacetate that can be used for gluconeogenesis, and guess what gluconeogenesis uses? NADH. You need NADH to make glucose. So consequently, doing this and then shoving this guy, oxaloacetate, out to make glucose makes very good sense for the cell. Does that make sense? How's everybody feeling? How are the note cards coming along? Anybody got theirs filled out yet? Nobody ever does it this early. That's why when people ask me to have a note card early, it's like, why? Why do you want a note card early? All you're going to do is lose it before you get there. People lay out their note cards and plan it accordingly. So, yeah, so good for that. How are you feeling about the exam? Are you anxious? You feeling better about this one, or more anxious, or less anxious than the last exam? Way more, way more. It's like me, I weigh more every day, it seems like, but bad joke. Well, this is an exam. Keep it in perspective. We've had 60% of the points in the class so far, and we only have 40% more on this exam. So while it's more than you've had on the other two exams, it's not 60% of your grade. It's 40% of your grade. I hope that helps. Panic look there, right? I was talking to somebody the other day, and one of the things I regret that I haven't done over the years is I never created a class on grading, because I find that students and even faculty don't know a lot about grading, and grading is really interesting and an important phenomenon, okay? So grading, there's a lot of ways of grading that you've seen. 90, 80, 70, 60 is one way of grading. Curving is another way of grading. It's the curving that people never seem to quite understand, which I find unfortunate. But one of the things that I've seen over the years, and I've followed this for many years, especially as old as I am, is looking at the performance on the final exam compared to the first two exams. And there is a very strong correlation with grades with those. You do see some changes, yes. Some people make that magical jump, and there's not very many that do that on the final exam. Some people have very bad performance, and I would say those two are roughly equal. But the vast majority of people, by a long ways, make just about the same region as what they made before. Now, the percentage may vary because the class percentage varies. But if you look at a person's standing relative to that class average, it really doesn't change that much. So I tell you that to encourage you, or at least to keep you from freaking out too much, that you got a bloody nose, is that what happened? Yeah. Did she punch you, or? Yeah. I say that so you don't freak out too much. It's not that big of a deal, to be honest. I know your grades are important. I don't mean to say they're not important, but I think that you will probably do about what you've done, okay? The other thing I tell students, and I've told a few of you who come to my office about this, is that the very best thing that you can do for exam anxiety is good exercise on the morning of the exam. Very good, strenuous exercise. And that's because what exercise does is it breaks down cortisol, the stress hormone. And the stress hormone was designed to help your body to escape danger, which means it diverts blood flow to your muscles so that you can run away from a grizzly bear that's chasing you. When it diverts blood flow to your muscles, it takes it away from your brain, which is why you don't think as well when you're stressed, because your brain isn't there thinking about Einstein's E equals mc squared equation. When a grizzly bear is chasing you, the only thought your brain has is get my ass out of here, right? So the way you beat stress hormones with exercise. So go out the morning of your exam and break up your cortisol by exercising the heck out of it, and you'll find it will make a difference for you with respect to your exams. Guarantee you. A lot of testimonials from students that really helped them. Now I'll shut up. This is the Sigma Delta Omega row here, right? So the two of you have Sigma Delta Omega shirts on. Are you Sigma Delta Omega? No, but these two are, so yeah. Okay, any other questions? Yes?
Mine's a quick random one.
Okay, a quick random one.
Yeah. So VLDLs and LDLs,
Yes?
do they have the same density of fat?
Do VLDLs and LDLs have the same density of fat? I don't know what you mean by density, but they have...
It was a question on your past exam.
What past exam?
The midterm two. Would
Um,
you like me to read you the question?
Read me the question.
Okay. So it was number 2. LDLs have a higher percentage of fat than VLDLs.
Okay, so percentage is different from density, right? So when you say that's why I say I wouldn't use the term density, because density implies something else. Density is weight divided by volume, right? So, and we talk about percentage, all right? Then percentage of fat within an LDL is going to be lower than it is in a VLDL, because the lipases have acted on the LDL to get it on the VLDL to get it down to an LDL. That makes sense?
I guess so. I just think of very low density as having less because it's lower. So do I
Okay.
have that switch?
Well, very low density is referring to the fact that it is less dense. But what makes it less dense? More fat, right? So you're losing fat. But don't think about it in terms of density. Think about it. So I describe the VLDL as a big Oreo cookie, and you're eating away the center of the Oreo cookie. The only thing that gets eaten out of any of these complexes is fat by lipases. So if you go from a chylomicron to a chylomicron remnant, the difference is that a lipase has taken some of the fat away from a chylomicron to leave you with a chylomicron remnant. The VLDL is the biggest complex made by the liver. So that's the thing that's released by the liver that says the body needs this stuff. So it's going to be full of fat, it's going to be full of vitamins, it's going to be full of cholesterol. But the vitamins and the cholesterol can't be touched by the lipases. So the only thing that can happen to convert a VLDL to an IDL is a lipase takes some of that fat. What converts an IDL to an LDL, a lipase takes some of that fat. So each step you're losing some of that fat. That make sense?
Yeah.
Yes?
Aren't LDLs the highest percentage of cholesterol?
LDLs have the highest percentage of cholesterol for just the reason I stated, because as the percentage of fat goes down, the percentage of fat-soluble vitamins and percentage of cholesterol is going to go up. Yep, absolutely. Right? Yes?
Can you go over like the hierarchy of prostogens and prostaglandins and prostanoids? I think I'm just getting them all.
You're just getting prosted out right there, right? Okay. So, prostanoids and prostogens, I would use those two terms interchangeably. Prostogens and prostanoids. Prostaglandins are a subset of those. So prostaglandins are one set of molecules. What are other molecules that are prostogens or prostanoids? That would include the thromboxanes and the leukotrienes. Actually, leukotrienes, I didn't group, let me back up on that. I didn't group leukotrienes in the prostogens in what I talked about. I described the leukotrienes as the 20. What's the word for 20? Not arachidonic, no. The I can't think of my own word here. Sorry. Pull it up here. The what? No. There's a word for 20. I can't think of the word for 20. Eicosanoids. There it is. Eicosanoids. So eicosanoids means 20.
So in the eicosanoids, there are prostogens and leukotrienes, those are subsets, and then in the prostogens, which are the same as prostanoids, there are prostaglandins, thromboxanes, and prostacyclins.
Yes, exactly. Very good.
There
Yes.
was one enzyme, I think, in nucleotide metabolism that was activated by phosphorylation. Which one was that?
So the question was there an enzyme in nucleotide metabolism that was activated by phosphorylation? Not that, no.
Was it in?
So there was one that was in, you're going to call in my head now. the fatty acyl-CoA, I'm sorry, the triacylglycerol lipase is activated by phosphorylation that's initiated by the epinephrine system.
Are most lipases activated by phosphorylation?
That's the only one that is. That's the only regulated enzyme of fat breakdown, and it's activated by phosphorylation. It's the only one. So the diacyl and the monoacyl are not. Yes?
Did we talk about phospholipase C?
Did we talk about phospholipase C? We did not talk about phospholipase C. Would you like me to?
No.
So, okay, well, I'm just going to throw this back at you as if I were asking you an exam question here. So if I said phospholipase C, tell me what it does. What would it do? What would it act on? It's going to break down phospholipids, and most likely, what would you suspect it would work on? A glycerophospholipid, right? Okay, because a lipase is going to be something that's going to work on a fat, and so the closest thing related to a fat would be a glycerophospholipid. A sphingolipid like sphingomyelin that contains phosphate would not be a fat molecule. So the most likely thing that a phospholipase C would work on is a glycerophospholipid, and in fact that's exactly what phospholipase C does. Phospholipase C, again, just because you've asked the question, not because you need to know it, is an enzyme that's important in the phenomenon of signaling. And so what it does is it clips the phosphate off of a glycerophospholipid, okay, and leaves behind a diacylglycerol as a function of that. Okay, and it's a little bit complicated after that, but that's what it does. More than you wanted to know. It's something I would have talked about. I would think Dr. McFadden would have talked about it last term. Did you guys not hear it in 450? Phospholipase C? Okay. Yes?
Mine's regarding vitamin B12.
Vitamin B12, yes.
It said that propanoic acid requires vitamin B12. but
Propanoic acid metabolism does, yes.
Okay. So then it also said odd-chain fatty acid. So any odd-chain fatty acid or just propanoic acid?
Okay, so odd chain fatty acids, when you break them down, give rise to propanoic acid.
Okay.
Okay, so propanoic acid is a 3-carbon, propanyl-CoA is a 3-carbon CoA. So if you started with a 15-carbon CoA and you take 2 carbons off at a time, you're going to go 15, 13, 11, 9, 7, 5, 3, and you're a propanoic acid. So anything that has an odd number of carbons is going to end in propanoic acid. Then that has to be handled. Everything else in that 15-chain carbon fatty acid can be broken down by beta oxidation because it can still clip off 2 at a time. It only stops when it gets down to the 3-carbon piece. That's when the cell's got to have something for it. Okay? Well, I'm beginning to think we've reached the end. People are probably getting hungry like I am, if I'm guessing right. So let's call it an evening. I will be in my office on and off, so if you want to meet with me, send me an email. I'll be happy to arrange a time to meet with you. I should be around my office in the morning. I've got to meet with my tax person, so morning may not be a good time to catch me tomorrow, but I think mid-afternoon tomorrow would be a good time, and I can always try to schedule a time with you as well. So study hard, get the note cards together, and I'll see you Thursday morning at 9:30. Take care.